Good morning, good afternoon, good evening, everybody. I'm Carla Roslin, and it's an absolute pleasure to introduce our global immunospeaker today, Dr. Kees Murre. Kees did his undergraduate in chemistry at the University of Amsterdam, and I learned that he was inspired by Ludwig Boltzmann, who was instrumental in proposing a statistical explanation to the second law of thermodynamics. Maybe, Kees, we look forward to learning more about this inspiration that you have while you were growing up and taking your first steps in science. Kees then continued his studies and did his PhD in a joint program between the University of Leiden and Harvard Medical School. During this time, he trained with John Seidman at Harvard Medical School in the Department of Genetics. Just as context, this was a very, very exciting time. This was back in the 80s, where the T-cell receptors and their genetic rearrangements were being discovered. These were Kees' first steps, I think, in genetics and immunology. And his PhD work significantly contributed to this field, identifying gamma change genes that undergo somatic rearrangement in human cells. He then did his postdoctoral work with Dr. David Baltimore at the Whitehead Institute, where he continued to do groundbreaking research in gene regulation, specifically contributing to the discovery of the helix-loop-helix motif. As I understand, this fundamental discovery really launched Kees' independent career. He joined UCSD in 1990, quite some time ago, where he has been there ever since and raised to the rank of distinguished professor. His laboratory has made fundamental contributions to the understanding of helix-loop-helix proteins in lymphocyte development, and I believe the focus on molecular mechanisms on the regulation of genome topology and gene expression has inspired him to also study how polymorphomononuclear structures emerge, something that is very much characteristic of cells like neutrophils, but whose underlying mechanism was really unknown until very recent studies from the Murre lab. I'm sure we're going to learn more about this today in his seminar entitled Building Polymorphomononuclear Cells. On a more personal note, I have been very fortunate to get to know Kees a little bit closer. I think he's one of those colleagues that one is very grateful to have in the field. I very especially admire his curiosity, his boldness, and his rigor. And on top of that, he's a delightful human being. So Kees, thank you so much for accepting our invitation to present in the Global IMMUNOTALK. It's a delight to have you here with us.
Thank you, Carla. Thank you for your comments.
Thank you. So before we get started, we always love to know more about our speakers. And I think in very much alignment of what are your characteristics, we would really love to know what strategies do you use to stay innovative and motivated in your research?
Yes, thank you, Carla. But let me first address the first question.
The first question, absolutely.
About the Boltzmann thing.
Yes.
So I was an undergraduate in chemistry, and we were required to take a course in statistical mechanics. That was a requirement in those days. And to my surprise, the professor decided to do an oral examination. And so I was, of course, a bit nervous about this. So, but then it started out like a conversation, and I learned more in those 15, 20 minutes about statistical mechanics than in the whole course. And so the professor was really influential, because that's how I try to teach as well, to have a conversation with my students. but then, of course, then he said after 15 minutes or so, he says, well, that's it. Let's talk about Boltzmann. And so he told me a little bit about him. And, the amazing impact he had, that he generated this field entirely by himself. And it turned out that in the next 100 years or so, nothing has changed since the Boltzmann proposal on statistical mechanics, that the idea that the movement of atoms and molecules defines the property of matter. But at the time it was very controversial, and because the philosopher Ernst Mach, who was actually located in the same place as Boltzmann was, he always postulated, and he had major influence on the field in those days. He always says, well, atoms, have you ever seen one yet?
Right.
Of course, they couldn't detect them. And so Boltzmann was quite lonely in his field, even though, of course, it ended up that atoms indeed did exist. And that was proven maybe 20, 30 years later by Max Planck and Einstein. So, but the sad thing was that he was already prone to depression, and this major opposition affected him deeply. And of course, in the end, his life ended in tragedy. Can you imagine that? One of the greatest ever? And he was bullied by so many of his colleagues because of a theory. He was way ahead of his time, in fact.
Right.
And not only that, someone pointed out recently that Boltzmann was, he was also important as it relates to molecular biology, believe it or not. So when he was a professor in Vienna, he mentored Lisa Meitner. And of course, he's the great nuclear physicist who should have gotten the Nobel Prize, but didn't. And then Lisa Meitner, in turn, mentored Max Delbrück for a couple of years. I don't know for how long. And of course, Delbrück was instrumental as it relates to the beginnings of molecular biology. Right. So you can see that a lot of things started with Boltzmann. Yes. And there is a very good biography written by him, written about him. And I recommend people to read it. It's very inspiring. Thank you so much. Yeah. So that's the Boltzmann response.
Okay, great to know more about your perspective on this. Thank you so much for sharing that. I'm sure many of our attendees will be inspired to learn more about this.
Yeah, I don't pretend to have
innovative.
I'm sorry. Yeah, I don't want to pretend that I have a deep understanding of statistical mechanics. That's not true at all.
Okay.
You know what I mean?
Appreciate that clarification, but I understand how sometimes, for those that are bold enough to come up with new concepts and ideas, they might not see it being accepted so easily or even in their lifetime.
Yeah, that's right. So now the second question. So where do I get my inspiration from? I think it's like probably true for most people. You get it from the conversations you have with your colleagues and your trainees. And in particular, my students and my postdoctoral fellows over the years, we had so many long conversations, and I learned from them, and really they had a major impact, of course, without them. And I was fortunate to have many very good students and postdocs. So then the second thing is, I was always interested in reading the history of science or history in general, and including what we just talked about, and reading about their lives and how they ended up looking or trying to figure out a problem that developed their curiosity. And I guess it's a matter of being curious all the time. Other things that really impacted me were the description of their works, and of course what comes to mind, that's not just me, but many, many before me, is this little book of less than 100 pages that Schrödinger wrote, What is Life? And where he compared the differences in structure of normal and aperiodic crystals. And he suggested that the chromatin fiber would be structured like an aperiodic crystal, like a masterpiece of embroidery, like a Raphael tapestry. And it was written so beautifully and inspired us to start thinking about chromatin motion and to track Ig heavy chain locus motion in live cells. That turned out to be much more complicated than I thought it would be. And so our contribution have been minimal so far. Then, other things that happens during, when you grow up and you're an undergraduate and you go to courses. And for me, I was a chemistry major, but sometimes I learned about biology, and I, at some point, noticed or was alerted to the fact that peripheral blood cells adapt these beautiful nuclear shapes, that differ greatly among each other. And, the images were very beautiful because they, light blue and purple bands, and the white Ginza staining, I think, are very easy on the eye. Beautiful, really. And so I wondered, well, and that's when I was an undergrad, well, where do they come from? And I didn't think about it much for a long time, but it always stuck with me. At some point, Hi-C was developed by Lieberman Eiden and Job Dekker. And I thought, well, now's the time to address this problem. And so we started in 2010 or 2011. And it took 14 years to get some idea what's going on. And that's what I'm going to talk about.
This is wonderful. Thank you so much for sharing this case. I think our attendees have gathered a little bit of a glimpse of how you are as a scientist. We'll learn more about it through your presentation. Really appreciate how things can take time. And it's the commitment, and the focus that move us forward. So we're all ears. We look forward to your presentation. Thank you.
Okay, great. Now I need to be able to move my screen, my images. How do I do that?
Let's see. Do you have in the bottom?
Oh, I should share. Is that what I should do?
Let's see. Let's try. Yeah.
But I cannot move it.
You cannot move it. Okay. Okay. Maybe can you stop sharing and share your screen again? Yes, please. Let's see. I can see that. Let's see. Can you pass it either with the computer? Perfect. That works. That works. Do you see it on your side?
Yeah, I have to do it in a certain way, but
fantastic. It works on this side. This is great. Thank you.
Okay. Let's get going. Okay, what I'm going to talk about is about the morphologies of the nuclear structures in specialized kind of cell that's called a neutrophil. The work was started maybe about 14 years ago by Yina Zhu and followed up by Matt Denholtz, who have since left the lab, but they really provided the foundation of the studies that I'm going to talk about. Indu Pata then continued the work that was started by Yina, and she was the first to show that diminished loop extrusion instructs the assembly of polymorphonuclear structures. Arpita Nidhi and Akira also contributed, and I'll mention their contribution during the course of my talk. Tegan and Hanbin also made contributions. The bioinformatics was done by Ming Hu and Shreya Mishra at the Cleveland Clinic Foundation. And Christine Chang analyzed some single-cell RNA-seq data for us, together with Mariam. Yuan Hu did the typing of the different polymorphonuclear structures. He's in the Department of Hematology at UCSD. And Sarah and Z at the LGI helped us with determining the nuclear volume of the differentiated cells. Now, as I mentioned earlier, when you take a look at peripheral blood, what is immediately apparent that many of these cells adopt very peculiar structures. And these are polymorphonuclear in nature, and you can see one here. It's probably a neutrophil where we have the distinct lobes that are often connected by linkers. Then there are other kind of morphologies. I think this is an eosinophil that is bilobular in nature. Here we have presumably a basophil-like topology. So in other words, a substantial fraction of the peripheral blood cells adopt unique structures, and the question is, how does that work? During human hematopoiesis, you can easily identify these different structures. For example, here we see a promyelocyte. This is a picture taken by Huan Yuan. Here is a myelocyte nuclear structure. Here is a nuclear morphology that is assembled in the metamyelocyte, another progenitor or intermediate progenitor of neutrophils. Here are band neutrophils. And then here is the more differentiated neutrophil, in humans at least, that shows segmented nuclei. So our goal was to figure out how this works. It's really a problem of nuclear folding. The neutrophils were identified by Mechnikov in the late 19th century or so. He's the father of innate immunity. Here he is, he was Ukrainian, or he had a Moldavian father and an Ukrainian mother. He was good friends with another giant of humanity. It's Tolstoy. And I think it's rare to see photographs where you have two jewels of humanity in one photograph. Eli Mechnikov made drawings of the things he saw under the microscope. And here you can see one of his beautiful drawings. This is a neutrophil. And there's actually a plasma cell. It's with a nucleus and a polarized orientation as it relates to the nuclear lamina. You know, Mechnikov actually described the first one, the multilobular organization, as that neutrophils are distinguished by having a nucleus which, although single, is divided in yet continuous, yet divided into several lobes, and here we have two of those lobes shown. And so then the first question that comes to mind is that every immunologist, of course, wants to know what is the purpose of these polymorphonuclear shapes. Why do cells assemble into these distinct structures? Well, there are two possible scenarios. The common accepted view is that the polymorphonuclear structures allows cells to squeeze through very tight interstitial tissue spaces. But there is another one, and I think that's becoming more and more of interest these days, and that is that the assembly of these distinct lobes reorganizes the cytoplasmic architecture and may allow for the spatial enrichments, let's say, for example, of granular bodies in neutrophils to attack the evading or digested phagosomes. Now, how this all works out, we will have to see, but it seems that these polymorphonuclear shapes, likely that they will have distinct functions. So we wanted to know how these polymorphonuclear shapes assemble. And we used a model system that was established by Mark Camps. I worked together with Mark when he was still at the White Hat and when I was also a postdoc. And he was a very nice colleague. And then he became a professor at UCSD. And when he retired, he gave me his ECOM-G cells. It's a system that allows mononuclear cells to rapidly differentiate upon withdrawal of estradiol into hypersegmented neutrophils. So that's the system. You have mononuclear cells, and these cells grow in the presence of estradiol that activate a proto-oncogene, in this case E2A-PBX. If you knock out E2A-PBX by withdrawing estradiol, these cells differentiate into becoming polymorphonuclear neutrophils. This happens in 4 to 5 days. So you can follow it and you can see, you can detect the intermediate polymorphonuclear structures. Hina did a high-C analysis, maybe already more than 10 years ago, and then compared neutrophils versus progenitors. And this is shown for chromosome 17. So high-C basically allows you to detect the frequencies of genomic interactions, and you can compare them, in this case, neutrophils to progenitors. That means the red indicates more interactions in neutrophils as compared to progenitors, the blue indicates fewer. This is the chromosome axis, and you can see that the entire chromosome 17 locally is depleted for intrachromosomal interactions. So a decrease in local TADs, topological domains, as indicated here. So that is a significant decrease, and this is true for all chromosomes. Now, you can also see that the interactions across vast genomic distances along the chromosome is enriched in neutrophils. So we see the decline in local interactions, whereas long-range interactions or remote genomic interactions are enriched. You can plot the frequencies of intra-TAD domain interactions. So these are rosette-like structures. And then within these rosettes, one can compute the frequencies of interactions. And that's done for progenitors. And you can see the average is shown here, or the distribution. You can see there's a significant decline of intra-TAD domain interactions in neutrophils. So overall, it seems that neutrophils seem to be declined in the density of local loops, but they are enriched for long-range genomic interactions. Now, of course, loops are generated by loop extrusion. And for those that are not familiar with it, there is this diagram that I obtained from a paper. I don't remember it, but I should have referenced it. Anyway, here we have two CTCF sites. So these are the binding sites that anchor the loops. And here we have a motor called the cohesion complex that extrudes loops until, and it's shown here, the beginning of the loop extrudes further, reels in chromatin until the base of the loop, the two anchor CTCF site, two paired CTCF sites are reached. So then we have a loop here, and then multiple loops can assemble into a bundle of loops or rosette or a TAD-like structure. Now, this is mediated by a bunch of proteins. Particularly important is RAD21. It's the motor that extrudes the chromatin through this cavity of the complex. RAD21 is activated by a loading factor, NIPBL, and I'm going to talk about this extensively. NIPBL activates RAD21 and the loop is formed. It also involves the expression of other components, and two of them are shown here. So the first question is, well, we see a decline in the number of loops in neutrophils as compared to progenitors. How does this relate to the expression of factors like NIPBL or like RAD21, SMC1, and SMC3 during the course of neutrophil differentiation? Indu plotted the RNA-seq patterns for progenitors and neutrophils for several of these loop extrusion factors. And one of them is indicated here, SMC1A, SMC3, RAD21, and NIPBL. Now let's start with NIPBL. You can immediately see that NIPBL is highly expressed in progenitor, but it declines in differentiating neutrophils. And the same is true for RAD21. It also declines. There is still activity present, though. It's not completely abolished, so we prefer to say it is diminished. And the same is true for SMC1 and SMC3. So overall, the conclusion is that loop extrusion activity declines during the course of neutrophil differentiation. So what we then did, we focused on the protein that activates loop extrusion, and that's NIPBL. It activates RAD21. So we thought when we would acute deplete NIPBL in these progenitors, so by blocking or diminishing loop extrusion, would we possibly induce a neutrophil specific cell fate? That seems far-fetched, but it was in the middle of COVID, and We did have nothing else to do, I guess. And so that's when we started this experiment. And we didn't only check NIPBL, we also checked MAU2, which is a partner of NIPBL, as well as RAD21 and a bunch of others. So, but we started with NIPBL, and that was a good decision. More luck than wisdom, to be quite frank. So now we have the system. We have mononuclear ECOMG cells that self-renew indefinitely in the presence of estradiol. We inserted into the endogenous loci a NIPBL FKB12 degron that was fused to EYFP. Now, DTAC13 is a cell-permeable compound that will bind to FKB12 and then will rapidly degrade NIPBL. So that will undergo rapid acute depletion of NIPBL abundance in these progenitors. And then the question is, does this allow the differentiation of a mononuclear progenitor-like cell into a differentiated hypersegmented neutrophil? We first checked whether the system worked when these cells were cultured in the absence of DTAC13, and in the presence of DTAC13, you can see NIPBL is completely abolished. Here is the first experiment that we did. Here is a right Giemsa staining of the ECOMG progenitors. Sometimes we call them neutrophils progenitors, sometimes myeloid progenitors. They are mostly mononuclear, as you can see. But there are some kidney-like, bean-like shaped morphologies. Now, it became immediately apparent after 3 days or so that these cells adopt polymorphonuclear structures. You can see the lobular structures here and here. There are also many cells that adopt a bean-like, kidney-like morphology. We have ring-shaped or donut-like structures. And these are all kind of define the many different cell types that accompany neutrophil differentiation. So that was a remarkable result. I remember when we first did this experiment and we added DTAC13 to these myeloid progenitors and we looked at the cells just under the microscope, we could already see it. The cells became smaller and they were attaching to the plates, so we immediately thought, wow, these could indeed be neutrophils. Here we are zooming in onto these neutrophil structures. You can see these are the ring, the donut-like morphologies. Here we see a hypersegmented neutrophil morphology, multilobular structures that are arising upon depleting NIPBL, so basically removing the loops from a large part of the genome. Then we thought, well, if NIPBL works, then its partner should work. The heterodimeric partner of NIPBL is MAU2 in the loading process. And here you can see that it seemed to actually even work better. You can see many of these cells adopt these wonderful polymorphonuclear structures. I always like to look at them. They never get boring. There's so much heterogeneity among these structures. Even within the population and during the course of the loss of loop extrusion. Here we're going to zoom in again on several of these, but you can see nicely these lobular structures that are connected to one another via these linkers. I think these are chromosomes, but one or a pair of chromosomes. But we still have to figure that one out. Here we're knocking out both MAU2 and NIPBL. And now it's, the majority of these cells adopt polymorphonuclear lobes, as you can see here, all over. Beautiful multilobular structures. Hypersegmented. Yuan Huang, our collaborator in hematology at UCSD, quantified this. He types all the different cells. So this is about 400 cells. 26% are now hypersegmented. And with many, of course, in an intermediate conformation. Indu decided then that she wanted to follow the assembly of these polymorphonuclear structures because it's an interesting problem of morphomatics, to try to identify a folding program that underpins the assembly of these many different nuclear morphologies. After 6 hours, we don't see much of a difference of removing NIPBL. After 9 hours, we can see some minor changes, but they become more apparent after 18 hours. After 24 hours, we can really start seeing the shapes that are associated with more differentiated neutrophils. So it seems to be sequential and ordered. But it's also quite heterogeneous, so many different shapes are assembled. And of course, we need to know how this actually works in detail so we can have an understanding of the assembly of nuclear structures, nuclear morphologies in vivo. Now, of course, everybody asks, well, are these really neutrophils? They are assembled as polymorphonuclear cells. So we checked by flow cytometry. We measured for CD11b and Ly6G expression. Ly6G expression indicates differentiation towards the neutrophil lineage. And you can see in the absence of DTEX13, it looks like this. Majority of cells is low for CD11b and mostly negative for Ly6G. Then after 72 hours, when we remove loop extrusion by adding DTEX13, you can see these cells readily differentiate to become Ly6G, CD11b double positive. And the same is true for MAU2, although the induction of a neutrophil-specific program is less robust when we just deplete MAU2. The order of the assembly of polymorphonuclear structures upon depleting for NIBL is also closely associated with the induction of Ly6G. After 6 hours of depletion of NIBL, we don't see not many neutrophils, or 0. The same as 9 hours. 18 hours, we can see them appearing. And then they accumulate after 24 and 72 hours. We have a significant fraction of the cells that become Ly6G, CD11b positive. Now, the nice thing about the system is that it is so reproducible, it's very robust, it works pretty much every time, independently, whoever does it, because the results are kind of remarkable. Simply removing the loops activates a neutrophil-specific gene program and initiates the assembly of polymorphonuclear structures. Now, then the question arises whether this process is reversible. So we can now, basically the idea is we culture the cells in the presence of estradiol that keeps them presumably in a multi-progenitor-like cell state. Then we add DTEX13 that we know instructs the assembly of polymorphonuclear structures. And then the question is whether we can go back to here by removing again DTEX13, but keeping it in estradiol. And so the question is whether these will go into this way or whether they will do whether they will become remain polymorphonuclear. In other words, is it reversible? So we counted the number of round cells, mononuclear cells, in the presence of DTEX13, and that is shown here. The number of round cells, of course, is not that high as in the absence of DTEX13, since they have lost loop extrusion activity and they are differentiated. The number of segmented cells, of course, is increased after you withdraw, after you remove loop extrusion, when the cells are cultured in the presence of DTEX13. Then when you remove DTEX13, you can see a substantial fraction of cells goes back to becoming mononuclear again. So there is some reversion taking place, but not all of it. Whereas on the other hand, the number of hypersegmented cells declines, as what you would expect. So there is indeed a trend for, we think these are mostly the intermediate, that are not completely terminally differentiated, that revert going back to a mononuclear-like cell state. But again, it's incomplete, and it may reflect the state of the differentiation state in which the cells are located or positioned. Now, as you would expect, when you withdraw NIBL in these progenitor cells, they activate, of course, a neutrophil-specific gene program, and they induce the expression of hundreds of neutrophil-specific genes. Genes that are involved in neutrophil physiology, like phagocytosis, they are all coordinately activated. The big question still is whether they also accumulate in granular bodies. We are looking at that together with at the La Jolla Institute for Immunology. So we need to know more about the cell biology that's associated with the differentiation from of progenitors into differentiated neutrophils by removing loop extrusion. You would expect, of course, many transcription factors that are associated with neutrophil differentiations are becoming activated, including the members of the CBP family and SPY1. NFIL3 and neutrophil-specific transcription are closely associated, I should say, because none of them is totally restricted to neutrophils, is also activated, as well as IRF4 and IRF5 and IRF8. Now, many of you will say, well, particularly immunologists, well, this is in the cell line, what happens in primary cells. And so what Indu did, she depleted NIBL in GMP-like cells in vitro using RMP electroporation and using CRISPR. And that really worked well by targeting the NIBL dimerization domain with MAO2. So these is the control. These are cells that were not electroporated by ablating NIBL expression. Here is the GMPs, and these are the more differentiated neutrophils. There are some present. Now, when we remove NIBL expression by targeting the interface between NIBL and MAO2, now you can see the GMP population has declined, and the neutrophil, the mature and immature, as well as transitional neutrophils, these populations have been greatly increased. Myelocyte populations are also elevated upon depletion of NIBL. It's kind of consistent with what we see with the ECOMG experiments. So now we have two observations. Diminished loop extrusions instructs the assembly of polymorphonuclear shapes, and diminished loop extrusion is sufficient to activate a neutrophil-specific gene program. And now the question is, now we need to, because loop extrusion is obviously involved, so we need to understand the changes in chromatin folding, how those changes could possibly lead to changes in nuclear morphology. I think that's kind of the key question, or the most intriguing question at this point. Of course, there are many genes going to be activated. I'm going to talk about this slowly to you in a minute. So what we have here is a high C matrix, and you see the contact frequency plotted as a function of genomic distance. And what the red indicates NIBL removal. This is the wild type, normal levels of NIBL. You can immediately see when you remove NIBL, you lose, there's a decline, steep decline in local genomic interactions. So these are the TADs, the rosette-like structures that are assembled all along the chromosome. In the absence of NIBL, on the other hand, genomic interactions that span vast genomic distances, over 100 megabases or so across the chromosome, are highly enriched. So there's a very dramatic change in nuclear, in chromatin folding and genomic interactions. It's very similar to what people have seen before by Budinger in particular, and Jesse Dixon in a paper that's on the bioarchives for other cell types, including embryonic stem cells. So, and initially by Francois Spitz in liver-like cells. So that is not as such not novel. Now, the first thing we did is to compare the chromatin folding pattern in the ECOMG that were depleted for NIBL, so in the presence of plus DTAC versus minus DTAC, and then compare it to the same a differential high C matrix when we're comparing neutrophils to progenitor cells. So these were differentiated by simply withdrawing estradiol. You can see when you withdraw a loop extrusion, the pattern of genomic interactions across this chromosome, chromosome 17, is very similar as when you plot the high 3 matrix for chromatin interactions for chromosome 17 when comparing progenitors to neutrophils. So the degree of loop extrusion, of course, is much more prominent when you completely wipe out loop extrusion by removing NIBL, as compared to what happens during normal neutrophil differentiation, where you can clearly see a substantial decline in local interactions, whereas remote genomic interactions are elevated. Now, first, a brief moment, and I'll discuss very briefly and quickly how it may affect gene expression, the removal of loops. Here, for example, you have a particular gene with a promoter that's located in one loop, and here we have an enhancer in the wild type situation. The enhancer is insulated from the promoter and by another loop, and that prevents, of course, the activation of the promoter. When NIPBL is removed and MAR2, the loop is removed. Now the insulators are no longer functioning because the loop has been abolished. Now the enhancer can make contact with the promoter and activate transcription. That's one possible scenario. Another possible scenario is that you have a promoter located in one loop with an enhancer, and the assembly of the loop would place this enhancer in close spatial proximity from the promoter so it can activate transcription. If the loop is removed, then in 3D space, these things will be far apart and the enhancer can no longer contact the promoter. So one example of this is, for example, the MYC gene. You can see MYC in these progenitors is highly expressed. If you remove NIPBL, it's within 2 days or so, it's completely inactivated. It might be actually happening earlier. And that's accompanied by a dramatic change in chromatin folding or interactions. You can see this is the interactions in the progenitor state. They map closely to an enhancer region located here as revealed by ATAC-SEC. When NIPBL is removed, not only we lose transcription here, but this particular region, including CTCF sites and enhancer elements, is declining, and that then results in a loss of a number of these loops that we think then inactivates the c-MYC expression. And that's basically what you see here. You have an enhancer communicating with the promoter in MYC, separated by large genomic distances. That had been shown by many people before. And now the loop is gone, MYC is inactivated, and that then leads to the rapid G1 arrest of these differentiating progenitor cell. And that might be actually a key event in promoting the developmental progression of these progenitor cells into differentiated neutrophils. There are other mechanisms involved as well, like the cohesion is also important for the association of sister chromatids during cell cycle progression. And that's mediated, that is instructed by NIPBL in the G1 state. So if NIPBL is no longer present, that also, of course, allows to the G1 arrest. That may be why the whole system works so well. Immediate G1 arrest, induction of neutrophil differentiation and assembly of polymorphonuclear structures. Here is another scenario where we have NF-IL3, a transcription factor. It's not expressed in the progenitor, but it becomes activated after withdrawing loop extrusion, as you can see. Look at the scale bars. The activation is very dramatic. The removal of the loops is indicated here. There are many intermediate looping patterns present in the progenitors that prevent, we think, enhancers to interact with NF-IL3 promoter. And that then results in the activation of the removal of these insulator components, factors, I should say, CTCF presumably, allows the enhancer to become spatially positioned within close spatial positioning of the NF-IL3 gene and activated. And we think that many of these genes become activated in this manner. Now we can also check whether the process of induction of gene expression is reversible, and you can see this here. We have in progenitor cells minus DTAC, we have an ensemble of genes, 650 genes or so that are activated. And when you remove NIPBL, when you then remove DTAC, so you reactivate loop extrusion, you can see that the transcription of many of these genes goes down again. And the reverse is true for genes that are being repressed, like, for example, c-MYC. And that's shown here more quantitatively. Here are c-MYC levels expressed in progenitor cells. It's high, very high. Make sure that these cells self-renew. In the presence of DTAC, MYC levels drop quite quickly, dramatically, causes G1 arrest. And we think the initiation of a neutrophil-specific transcription signature When you remove DTAC13 again, MYC comes back up for a significant fraction of the cells. It's remarkably reversible, I think, for the cells that have not yet completely committed to the neutrophil cells fate. Now, I mentioned before that we have an enrichment for very remote genomic interactions, megaloops, in NIPBL depleted cells. That's what you can see here. The red is NIPBL depletion. And so we call these things megaloops. My friend Mike Levine names them metaloops. He identified them with a collaborator in the nervous system. So now the question is, what do these megaloops and metaloops really do? There are indeed hundreds of them present. So here's one, for example, that involves the NLRP3 gene, I believe it was, located on, I don't remember which chromosome it was. It's on some, oh, yeah, I don't remember. But anyway, the NLRP3 megaloops is assembled by interacting with another locus that is called SEC14L1 on the same chromosome. That interaction is absolutely absent in progenitors. It is highly enriched in differentiated neutrophils, and this spans about 100 megabase pairs of DNA. This is the differential high CMAP for that particular locus. So now we have euchromatic megaloops. They're associated with the anchors that are depleted for TATs. So the entire chromosome X is depleted for TATs. And as Budiger suggested, that makes the chromosome much more flexible. So paired elements that are separated by vast enormous distances can find each other. That normally would not be allowed when the chromosomes would be structured as stacked rosettes, which would make the chromosome relatively rigid. It's gene-rich at the anchors, and the genes at the anchors of megaloops, interestingly, are closely associated with a neutrophil-specific or an inflammatory-specific gene program, like toll-like receptors, components of the NF-kappa B pathway, and so on. And of course, as you would expect, they are highly enriched for neutrophil-specific or neutrophil-restricted, I should say, transcription factors, including P1. Also, Icarus and AP1 binding sites are highly present. We are also seeing a very high enrichment for interchromosomal interactions all across the genome when we deplete for loop extrusion. And here's one of such interchromosomal sites indicated here, involving the TREM1 gene involved in inflammation, as well as PTPRJ on another chromosome. This is chromosome 17 with chromosome 2, as well as the SPY1 locus. And each of these genes, like PRTRJ, is being activated, and TREM1 and closely associated with the induction of an chromatin accessibility at these sites. So we have also, of course, heterochromatic megaloops that are being generated after removing loop extrusion, and there are 150 or so present. They are gene poor, and we suggest they play a structural role. Perhaps they're involved in chromosome compaction that maybe allow the packing of the chromosomes into tight nuclear lobes. Here we have an interchromosomal. So now the question is, how do these things assemble? We have interchromosomal hubs indicated here. We have enhancers that are present here. This is one chromosome, and this is another chromosome. There's the enhancers. And then the idea would be there would be a nuclear condensate that would be assembled between the two chromosomes, allowing the interchromosomal interactions to form this transcription hub. May be involved not only in the activation of a neutrophil-specific transcription signature, but also perhaps involved in the assembly of the specific nuclear lobes. We think these interchromosomal interactions are going to be key. Megaloops may be involved in perhaps in the compaction of chromosomes, because the nuclear volume is greatly reduced, and they indeed are assembled by a cluster of enhancers that is present at the ends of the megaloop. So that's again shown here. So we have in progenitor cells, we have a chromosome, we have stacked rosettes. The chromosome is relatively rigid. It does not allow interactions to occur between paired elements that are separated by vast genomic distances. When the tads are removed or the rosettes are removed, the thing becomes much more flexible. So now paired elements involving paired genes, they can find each other more readily and assemble into nuclear condensates where transcription or activation, perhaps repression, is activated. And that's shown here, how these things then ultimately assemble into these hypersegmented structures. That's what we are really working on now, and that remains an interesting problem. You will ask immediately the question, well, what about human cells? So human neutrophils, are they also structured in a similar manner when we compare them to other cell types? And this analysis was done by Ming, and he obtained the data that was originally described by Peter Fraser. And it's plotted here. We're comparing here the intra-tad domain interaction frequencies of human neutrophils, so in human peripheral blood, to B cells, CD4 cells, CD8, erythroid progenitors, megakaryocytes, macrophages, and monocytes. You can see compared to B cells and the T cells and the erythroid progenitors, neutrophils are show a significant decline in intra-tad domain interactions. So this is a measure of loop extrusion activity. Also in human peripheral blood cells, it seems to be declining. Now it's particularly prominent in macrophages. So I predict that the macrophages would have a genome with highly packed TAT-like structure, rosette-like structures, relatively inflexible, an inflexible chromatin fiber. And the same is true for monocytes. But there's one exception, and that is number 7, and that is neutrophils compared to megakaryocytes. Now, megakaryocytes are interesting because they're also kind of polymorphonuclear. They also contain multiple nuclei in one cell. So we at some point will need to take a look at this, how that works in megakaryocytes. Very intriguing cells, in fact. But overall, I would say that diminished loop extrusion is a conserved feature of neutrophils. But one question, of course, is what happens to eosinophils and basophils. We're still looking at this. Now here we have a human chromosome 5, comparing human neutrophils to human macrophages. And you can see, again, this is depletion, local depletion of intra-tad domain interactions. The blue color indicates this for in neutrophils as compared to macrophages. So fewer rosettes along this thing. Whereas the rest of the long-range genomic interactions are enriched. So diminished loop extrusion that then readily results in the activation and repression of gene expression signatures that are associated with inflammation, a nuclear envelope associated proteins, transcription factors, and so on. There are many changes that are occurring, and we need to decipher which of these are the critical ones. Increased chromatin fiber flexibility to enrich for megaloops and interchromosomal context. And we suggest that specificity, lineage or developmental specificity of these loops are instructed by transcription factors that bind to the enhancers. We just published a paper in a review in Trends in Immunology, and one of the reviewers was so kind to redraw a diagram for us. He explained our results much better than we did. And so we included this in the review. We are very grateful to the reviewer. Here we have GMPs upon the loss of loop or declining levels of cohesion or loop extrusion. These cells will differentiate into neutrophils. We think similar mechanism might be at play in the differentiation of eosinophils and basophils. When neutrophils become activated, Matt Denholtz in my lab showed this already a number of years ago, they'll become mononuclear again, and cohesion is readily distributed all across the neutrophil genome. How this relates to the activation problem is kind of obvious because we see an inflammatory gene expression signature correlating with the assembly of de novo loops. And of course, the big question is how this relates to the instruction of netosis, an intriguing problem. Oh, and then finally, what I wanted to do always was to generate artificial nuclear structures. Because if you can manipulate nuclear morphology, perhaps we can manipulate neutrophil physiology, and by manipulating migration, for example. It may also allow the changes in cytoplasmic architecture to see how the whole cell biology is organized by changes in nuclear folding. And so we did this experiment. Nidhi Singh did this. And Nidhi did a number of the experiments that I forgot to mention. She is a very talented graduate student, visiting graduate student from Jagan Pomgubala lab. So Nidhi manipulated loop extrusion in differentiated neutrophils. And by doing so, she found that now we can generate indeed new nuclear structures. You can see here is a neutrophil with 1, 2, 3, 4, 5, 6, 7, 8, 9 lobes in one cell. Really remarkable. Here is another cell where she did it again. Now here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So more than 10 lobular structures. We also aim to generate elongated tube-like structures, like it's kind of shown here. It's not a perfect tube yet. We need to work on this. But here you can also see, oh yeah, here is another cell where we have the lobular structure continuous, actually connected by, I'm sure these are multiple chromosomes that make these distinctive morphologies. We have to understand now really what makes these structures, how do they get, these are the constriction points. I think these are the key points that determine the structure, because basically you have a loop here, giant loops, not a loop at the level of TADs. I was not really planning to discuss it in detail because we have no clue. I'm just speculating. Okay. So now we have, now we are thinking about the future, as if I have one, but so the new challenges that lie ahead are identifying enhancer codes that we think underpin the developmental specificity of nuclear shapes and nuclear deformability. We want to generate all kinds of different artificial structures, artificial nuclear structures on demand. So for that reason, we need to understand the a genetic code, a blueprint that underpins the structures of normal nuclear morphologies. We need to understand how nuclear shapes are assembled. It's a problem of nuclear folding, like protein folding, but in a larger scale, of course. And how does this relate to cytoplasmic architecture? But to summarize everything, what we know for sure, we don't know much yet. But one thing we know after the past 3 years or so that we have, well, past 13 years, but then as it relates to loop extrusion in the last few years, that the nuclear lobes that were observed by Eli Metchnikoff in the late 19th century, we think it involves the control of loop extrusion. It's certainly we can induce neutrophil differentiation and polymorphonuclear assembly by ablating or diminished loop extrusion activity, and we can now generate shapes that even go further than the shapes that normally are assembled during normal differentiation. All right, that's it. Thank you very much.
Thank you so much, Kees. It's always such a pleasure to listen to you. Thanks for sharing your fascinating work. And thanks to you and your colleagues for revealing such a beautiful molecular understanding of these profound changes that happen in the morphology and the function of polymorphonuclear cells. Really beautiful. So before we leave, I would like to remind our attendees how they can ask questions. We have a slide projected now where you can see that you can find a tweet in our Global IMMUNOTALKS account. Please reply to that tweet. Kees is going to use the ImmunoSpeaker account And we look forward to your questions there. And I would like to invite everybody to join us next week when we will have Dr. Wendy Garrett joining us. So thanks again, Case. Thanks for your efforts in presenting, and we'll stay in touch. Bye, everybody.
Thank you.